Greetings everyone, my name's Erik Wright and I'm joining you from the University of Pittsburgh. Today I'll be talking about community detection at unprecedented scales with ExoLabel. But before I begin, I'd like to point out that this work was done in conjunction with an excellent student in my lab named Lakshman. When we do comparative genomics, we usually start off with N genomes and then we do pairwise comparisons between them, resulting in N squared comparisons that we then put into some sort of sequence similarity network, resulting in gene groups that can be used in downstream applications. Today I'll be talking about this sequence similarity network step in the middle because it represents a bottleneck through this process. The N squared comparisons can be done distributed on a computing grid, but then this data comes back together to form a sequence similarity network, and that typically is done on a single computer, which makes this a rate limiting step in the comparative genomics workflow. When I talk about a sequence similarity network, what I mean is a network composed of genes as its nodes and sequence similarities as its edges. And what we'd like to do with a network like this one is be able to detect communities that are formed by sets of nodes, which are genes, that are closely linked, and disconnect weak edges between communities that are not strong enough to consider those communities part of the same community. This problem is called community detection, and there are several common algorithms. MCL is one that is regularly used in the comparative genomics field, but in other fields, Leiden and Louvain clustering are also commonly used. So, given a genome from an organism, which has thousands of genes in it, each additional gene that we add adds one edge per previous genome. And so the network size that we're working with scales quadratically with the number of genomes, presenting a very, very large problem to solve. And the problem with that is reading an entire network into RAM results in quadratic memory scaling, which is prohibitive on most machines. The reason being that the cost of memory over time has more or less plateaued, and not everyone has access to a high performance computer that has a very large amount of memory. But in contrast, hard drives are much cheaper and still falling somewhat in cost. And especially solid state drives, SSDs, are still plummeting in cost. So these present a viable alternative to RAM that can be used to process very large amounts of data. And for this purpose, we created a new program called ExoLabel, which is part of the SynExtend package in Bioconductor, and we're working on making it into a standalone application. The reason why we called it ExoLabel is a combination of label for fast label propagation, which is the core algorithm in which it's based upon, and exo because the data is stored outside of memory. We have memory scaling that is linear with the number of nodes, that is genes, and constant with the number of edges, because that information is stored outside of memory on the disk. And so ExoLabel presents a new option for clustering and community detection on sequence similarity networks that uses much less memory than existing algorithms. An example of ExoLabel's excellent memory scaling is shown here. What you're looking at is a log-log plot of the amount of memory used versus the number of nodes in the network. And these are for synthetic random networks with 10 edges per node. As I mentioned previously, ExoLabel's scaling is approximately linear with the number of nodes. And that's true for several of these algorithms, but some of them have quadratic scaling. More importantly, ExoLabel uses far less memory than existing algorithms at the same network size. And you can see that even for a very large network, which has 31 million nodes and 310 million edges, it's only using about 4.5 gigabytes of RAM on a single thread. And that's approximately 15 times better than the next best algorithm, which is Leiden. Normally there's a trade-off between memory usage and speed, but as you can see from this plot showing the amount of time it takes to detect communities in a network versus network size, ExoLabel is one of the most competitive in terms of speed as well. Many of the different algorithms have linear or linear rhythmic scaling, allowing them to process very large networks in reasonable amounts of time. But some of the popular ones, like MCL and HipMCL, have above linear and sometimes quadratic runtime scaling. ExoLabel was able to cluster the largest random network here with 31 million nodes and 310 million edges in only 46 minutes. So ExoLabel has excellent memory usage and competitive runtimes, but the question is, how accurate is it relative to other competitor algorithms? And to answer that question, I'll be showing you the results of 3 different network types. The first is synthetic networks that are created through LFR graphs where we know the exact answer. The next is simulated networks that mirror real sequence data. And the third is real networks where we have to use proxies for the correct answer. On these synthetic networks, what you can see is that ExoLabel, shown in purple, is competitive in terms of accuracy, but some of the algorithms, especially the slower ones, can beat it on these synthetic networks. Also, as the number of nodes in the network increases, ExoLabel more or less maintains its accuracy, whereas many of its competitors fall in accuracy relative to it. The problem with simulated graphs is that they aren't real, and in many cases, the assumptions of the simulation mirror the expectations of the algorithms that they're trying to compare. One of the big assumptions is that all of the edges will be present. In reality, things like blasts that are used in real networks miss many of the edges, especially the lower weight ones. And so, to look into this, we masked the lower weight edges at varying degrees to see how the different algorithms compare. What you can see here is algorithm performance relative to ExoLabel as different degrees of edges are masked. From 0% at the far left, which is the same as the data I showed you before, all the way up to 50% of the lowest weight edges. You can see that ExoLabel performs almost identically to label propagation in these simulations, which is what we expect because that's the algorithm that ExoLabel is based on. And it beats things like MCL using a high inflation parameter of 4.0 and leading eigenvalue. But the algorithms that beat it oftentimes get slightly worse and closer to ExoLabel as you mask more of the edges. And so this shows that there's slightly varying performance as you mask different numbers of edges, but for the most part, many of the algorithms behave similarly. Another issue with simulated graphs is that we don't know whether these LFR graphs that are very popular have the same topology as real networks. And so to construct something more like reality, we turned to a semi-synthetic benchmark. And here what we did is we used a known species tree from the OrthoMam database. And we simulated alignments from each of the genes in a seed proteome using the program ISMB. This allowed us to create a network with BLAST-P bit scores or PID. We then used these networks to compare the gene groups that we expect from the simulated proteome versus those that are found by each of the different algorithms. And what you can see in these semi-synthetic networks is that ExoLabel is competitive with many of the other programs. And in fact, some of the programs that do very well on purely synthetic networks actually do worse on semi-synthetic networks, such as Louvain and Leiden. InfoMap maintains its performance as one of the most competitive algorithms, but has the issue of poor scaling and poor memory usage. MCL, which is one of the most popular, also does well, just like ExoLabel, and you can see why it is very popular. We also compared using bit score in these simulations to using PID as the edge weights. And what you find is that bit score provides some slight advantage over PID, but both of them are much better than using unweighted networks. Semi-synthetic benchmarks are not as good as real data, of course. And so to construct a real benchmark, we used 4,422 NCBI prokaryotic reference genomes. We created a network with BLAST-P bit scores of each of the gene calls. And we analyzed the disjoint subsets so that all the different programs were able to compute the entire data set. Using sets of 50 real genomes, as shown here, we can look at mean within cluster PID versus mean cluster size. And what we would ideally want is for clusters to be large and contain very similar genes. So ideal performance is to the upper right on this plot. And what you can see is that ExoLabel produces clusters that are higher mean PID than that of MCL and are along the Pareto front, meaning that ExoLabel is one of the most competitive programs on real data. Each of these points represents 88 different graphs containing 50 genomes, on average with 145,000 genes and 1 million pairwise similarities. These are still very small graphs for a program like ExoLabel and only take a few seconds to compute. So we also looked at larger graphs, for example here with 100 genomes. And what we see is that, again, ExoLabel stands out above MCL in terms of its ability to create clusters that have higher similarity at the same cluster size. The parameter that's being swept here is what we call the self-loop parameter. And the self-loop parameter defines how similar outgoing edges have to be before we allow a network to be constructed, and is one of the features that we added to label propagation in order to make it work better on this type of data. MCL has a similar parameter in its inflation parameter that's being swept here. And you can see at any value of inflation versus self-loop, we still appear above MCL, meaning we get better clusters on real data. If we look at the runtimes on this real benchmark, MCL, as I said, for the smaller set only takes a couple of seconds, and for the larger set still only takes about 8 seconds. That's in comparison to the other programs, which are fast, but still are much slower than ExoLabel. ExoLabel is about 3 times faster than the next fastest algorithm. But these are still small data sets. So we want to try something much larger to see what the limits of ExoLabel are. For that, we clustered 16.2 million nodes, that is genes, and 18.3 billion edges, pairwise similarities, for all of RefSeq reference genomes, about 4,400 genomes. ExoLabel was able to process this in only 14 hours and 10 gigabytes of RAM, but required 600 gigabytes of disk space, about twice as much as the original data. This is what the resulting network looked like, at least a little part of it, where each of the different groups is represented by a color. And we can zoom in on different clusters, communities within this network, and look at what their functional annotation is. So here's an example of a couple of communities that are connected by weak edges, where the edge strength is represented by the thickness of the edges. These 2 different communities that ExoLabel picks out of this graph have different annotations, as you might have hoped for. And we see this pattern over and over again. Here's an example of 2 communities that ExoLabel splits out that have some connections, but those connections are fairly weak. And when we look at those communities, One contains proteins with domains of unknown function 523 and 1722, whereas the other contains just proteins with domain of unknown function 523. So this might be a reasonable thing for ExoLabel to have split apart. Here's another example. These two communities contain two different protein names, even though they have weak linkages between the two communities. So ExoLabel is approximately performing the way we would want it to if we were to do our network community detection by hand. But ExoLabel allows us to do it at a massive scale. Like I said before, it's available as part of the SynExtend package for R via Bioconductor, and we're working on developing a standalone executable. ExoLabel's as accurate as MCL, which is probably the next most popular program for community detection in comparative genomics, but it has a much lower computational requirement in terms of RAM and only requiring one processor. It's also even faster than alternatives, so there's no trade-off between memory and speed in the case of ExoLabel. With that, I'd like to thank my lab, and especially Aidan Lechman, who is the graduate student behind this work. I'd like to thank the NIAID for funding, and if you'd like to learn more, please go to our lab website at wrightlabscience.com. Thank you.